My name is Floriane and I'm in the last years of my PhD. I'm going to talk about teleost orthologs of human gene involved in a signaling pathway. Here we know that the creation of new genes is particularly due to whole genome duplication. And whole genome duplication have happened all along history, notably two at the time of the separation between invertebrates and vertebrates, then a third for all teleost fish, and a fourth for two distinct lead, calves and salmonid. It may seem that the number of genes is 2 times greater in teleost than the other vertebrates, but genome duplication are followed by a massive period of duplicate gene loss, resulting in genome like that of the zebrafish with around 26,000 genes, 26% of which are duplicate gene only. By comparison, human beings have around 22,000 genes. The same applies to species whose genomes has been duplicated 4 times. So we ask the question, why do all genes remain duplicate? What evolutionary pressure governs this return to the singleton or the maintenance of duplication? Secondly, in the literature, we have read that the maintenance of duplicate gene may be due to the sub or neofunctionalization of the duplicate genes or to the question of dosage. I'm interested in the question of dosage because we know that the subunits of multiprotein complexes need to remain in duplicate to maintain stoichiometry with their partner. And we also know that certain categories of function are enriched in duplicate genes, such as transcription factor or receptors of signaling pathway. So what about the quantity of teleost ortholog of gene product involved in the signaling pathway? Because a signaling pathway is a cascade of protein interaction. To answer this question, I use 47 human signaling pathway available on KEGG database, representing 3,000 genes and 2,300 unique protein interaction, and 63 teleost species, including 54 species with 3 whole genome duplication and 9 with 4 whole genome duplication. My method was to talk each of the genes, look at the quantity for each of the species. Here, A and B are in singleton and C in duplicate. And moreover, I also note the interaction where there are 2 of them, the first being of so-called respected stoichiometry and the second of non-respected stoichiometry because there are 2 times more C than B. Turning the results, first I look at the genes alone, and when we compare to the totality of the genes of the genome, our signaling pathway genes more often remained in the state of duplicate than the genes of the genome for the 3 species whole genome duplication, and in triplicate or more for species 4 whole genome duplication. I look at the integrating genes and the amount of each possible stoichiometry for each species. We see that for all the species, about 25% of the interaction are absent or partially absent. Then the stoichiometries diverge whether we are talking about 3 or 4 whole genome duplication. And for the 3 whole genome duplication, we have a majority of 1-1 interaction or 1-2 interaction, while for the 4 whole genome duplication, we are more on 2-2 and 2-4 interaction. What is very interesting is that despite a very distant evolutionary divergence, the profiles remain substantially the same. In conclusion, this work shows that teleost gene ortholog of human gene signaling pathway remain more in duplicate in the 3 whole genome duplication group and in triplicate in the 4 whole genome duplication group than the whole genome. Moreover, some genes involved in almost all pathways studied have been lost, and there are many protein-protein interactions for which the progression is not respected. And I'm waiting you in around the poster. Thanks.